So the next thing which you need to remember in this case is that prokaryotic cell, the nucleus which is without nuclear membrane, that is called as, yes, nucleus without nuclear membrane is called as what? Vaibhavi, what does it called? What does it call?
Prokaryotic cell.
Prokaryotic cell, but nucleus is called as, genetic material without nuclear membrane is called as? Genetic material without nuclear membrane is called as? Yes? Nucleoid, correct. It's called as nucleoid, and here it is a circular, big circular DNA. Understanding? Just big circular DNA. Then there are ribosomes. What is the size of ribosome in prokaryotic cell? Remember?
Yes, it is small.
Small, it is made up of two subunits, small and large. But what is size? What is its size? 70S.
70S.
And S stands for?
Sedberg's unit.
Correct, Sedberg's unit. Very good. Okay. Then the next thing is about plasmid. Okay. Now plasmid is very important thing to remember in the prokaryotic cell. So it is some extra DNA a prokaryotic cell has. Remember, only prokaryotic cell has plasmid. Okay. And it is a little bit extra DNA which help bacteria a lot. Okay. Remember, plasmid do extra functions, correct? Plasmid help in extra functions to bacteria. Helps in extra function, right? Extra functions like sexual reproduction, because bacteria otherwise don't do sexual reproduction. But if bacteria need to do sexual reproduction, it needs plasmid, right? Then another function is something called as antibiotic resistance. Antibiotic resistance is like, resistance against toxic chemicals. Okay. Resistance against means they get protection from toxic chemicals. Antibiotics are some toxic chemical for bacteria. Okay. About antibiotics, we will learn a lot afterward. Okay. But antibiotic resistance is also provided by plasmid. Okay. Otherwise, bacteria cannot have this, right? And then we even studied about inclusion bodies, correct? And we even studied about inclusion body. These are just food storage in bacteria, right? Those are just food storage particles in bacteria. Now, the next structure which we need to understand, next structure which we need to study is nothing but something called as mesosomes. Try to understand this mesosome, okay? Is screen visible to you, Vaibhavi? I did not ask. Forgot to ask. Is screen
Yes,
visible?
ma'am.
Vaibhavi,
Yes.
how you attend lecture on Wi-Fi or your 4G?
Wi-Fi. I have Wi-Fi at home.
Okay, then.
But today what happened? Morning on it rain, so network. I'm attending
From
Jio.
Okay. Your Wi-Fi is BSNL Wi-Fi?
No, Airtel.
Airtel Wi-Fi. Okay. It's not working today. Oh, so class also the
No.
Wi-Fi must not be working. Oh, okay. Still raining?
No, ma'am. Morning.
Morning
it was raining.
Okay. So, see, suppose this is our bacteria. Okay. Suppose consider right now which I am drawing is its this. Now, it's suppose it's cell wall, right? It is what? Cell wall. Suppose, huh? Okay. Inside the cell wall, there will be plasma membrane. Agree? There will be plasma membrane. Now, remember yesterday we compared compared bacteria like a single room. It's like a house with only one room. It do all the function. It is kitchen, means they cook food. They have bedroom, they study, everything. Everything is going on, but everything is going on in a single room, right? So they need more space. Agree? All the chemical reactions, Vaibhavi, are going on into the same place. Correct? So, and what is happening in the cytoplasm? What bacteria, what all organisms do is nothing but chemical reactions. Right? Chemical reactions. And those chemical reactions basically, remember, need place. Agree? Need space to complete them. Agree? They need space. Right? So to increase space, what bacteria do? This is its plasma membrane, that is cell membrane. Plasma membrane and cell membrane is one and the same thing. It produces extensions like this. It produces what? Extensions at a different location, at different shapes also. Sometimes just like this, sometimes very complicated shape. Sometimes the shape come near to the DNA. Understand that? Likewise. And these extensions of plasma membrane are called as mesosomes. They are called as what? Mesosomes. Remember, what are mesosomes? Mesosomes are extensions of plasma membrane, inside cytoplasm. Understanding? Inside cytoplasm, and they are of many different shapes. Understanding? Sometimes they are just tubules. These are like tube, correct? So called as tubule shaped. Okay, it is mesosome. Okay, sometimes it is having this complicated shape. Okay, this shape is called as lamellar shape. Complicated shape. Sometimes just just like this. Okay, it is called as a vesicle shape. Understanding? Vesicle shape. So it is having different shapes. Understanding? It is having different shapes. Clear? And but now, so mesosomes provide extra space for chemical reactions. Understanding? Reactions. Now one more function it do. Okay. See, these are functions. Here somewhere write functions. The first function is what? It provides extra space for chemical reactions to take place. Okay. Then secondly, it even helps in the cell wall formation. It even helps in cell wall formation. Mesosome help in cell wall formation. Thirdly, it even helps in DNA replication. You don't know what is DNA replication, just remember. I'll teach you DNA replication afterward. Okay. It helps in DNA replication. Okay. And fourthly, it helps to distribute DNA in daughter cells. Help to distribute DNA in daughter cells. Okay. Understanding? Oh, what happened? Missed call. Clear?
Yes.
Understood? Understood, mesosome? Okay. Now,
Yes.
The next few things we need to study. Then we will move to gram staining. Actually, I was waiting for Shrushti for gram staining. Gram staining is a little tricky part to teach. Okay. But till that time, I will teach you some small parts. Now, other structures also we see onto the bacteria. One structure present on bacteria. This is flagella. Okay. Now, yesterday only we studied some bacteria show presence of flagella. Some bacteria do not show the presence of flagella. Correct? It depends on bacteria to bacteria. Some bacteria show presence of flagella, some don't. But whenever bacteria have flagella, okay, see, whenever bacteria have flagella, it is motile bacteria, remember. Whenever bacteria have flagella, it is motile. No flagella, non-motile, right? No flagella, non-motile. Do you remember motile and non-motile meaning? Motile means it can go from one location to another location. It can swim, and non-motile cannot swim. Understand it? The bacteria called as typhoid bacteria, okay, typhoid bacteria, which causes typhoid, obviously, salmonella, it is having many flagella. Okay, it literally, when person is suffering from typhoid, those bacteria literally moving around in patient's blood, swimming in patient's blood. Okay. So, yeah, that's the, means I'm just giving you example. But on the other hand, if you consider curd bacteria, curd bacteria, which convert milk to curd, that bacteria cannot move at all. No flagella. Okay. Where you kept it, it will stay over there only. Understanding? Okay. So this green structure in this diagram, which you see, it is a flagella. Okay. Now, a flagella is made up of three structures in bacteria. Here, it show the attachment, correct? Attachment to the cell wall, not to the capsule. Attachment to the cell wall, and then it go inside the, means, what to say? Let's write about flagella. That will be easier. Okay. Let's write about flagella. There no enough space, correct? So let's write over here about flagella. Flagella, first thing is flagella present, it will be motile, flagella absent, non-motile, correct? Flagella absent, bacteria is non-motile, right? Bacteria is non-motile. Okay. Then flagella is extension from the, bacterium flagella is basically extension from plasma membrane. Okay, 1 second. No, that's not written as such in our book, but it will be. Okay, plasma flagella, generated or originated from plasma. Originated from plasma membrane in bacteria. Okay, that is one thing. The next thing which you need to remember, in motile bacteria, they may have single or multiple flagella, either one flagella or many flagella. Okay, one flagellum and many flagella. Okay, single or multiple flagella. That is one more thing you got to remember. The next thing you need to remember is, that flagella show three structures. Okay, flagella show three structures. Okay, one structure is obviously its basal body. Now try to understand. Suppose this is a plasma membrane, and above the cell wall, we will just write it. Okay, we will write it so you will understand it. Or you can, give different colors also if you understand that way. Okay, this is what plasma membrane. Okay, above that there is a cell wall, and above that there will be capsule. Agree? Above that there will be capsule. This we studied yesterday, right? There will be capsule. Okay. Now here there will be basement of the or basal body of the plasma, basal body of the flagella, right? Basal body. Okay, then it come out and form a hook-like structure, right? This will be hook. It take a turn when it come out of everything, correct? And then it is having that long, thin structure called as filament, right? That's why it is having this basal body, hook, and filament, these kind of structures. Understanding? Basal body is attachment. It come up, take a turn, it become hook, and filament is a thread-like structure. Understanding? Basal body, hook, and filament. Hook and filament. Understanding? Understanding?
Yes.
Yes, very good. Okay. Now, the other than flagella, like flagella is sometime present, sometime absent, remember. Okay, some bacteria have flagella, some don't. But all bacteria have one structure called as this pili. Called as these structures called as pili. Okay. When we talk about pili, no, pili are this extension of, remember, Vaibhavi, mesosomes were taking turn, plasma membrane was taking turn inside the cytoplasm, correct? But if you see the pili, that green color is the plasma membrane, it is taking turn outside. Okay, what it is doing? It is taking turn outside the cell, right? And form these projections outside the cell. These are the projections of plasma membrane, remember. See, pili are what? Projections of plasma membrane outside the cell. Okay, and what does they do? You know, they help the bacteria to attach for attachment. See, remember, let's write it. Let's write it. Okay, see, pili are what? Extension of plasma membranes. Extensions of plasma membrane outside the bacteria. Membrane, I'm just writing outside. Okay, outside. Multiple pili are there. So many are there. Many pili are present. And what does they do? They help, they help bacterial cell to attach to the surfaces. Cell for attachment. Okay, for attachment. Understanding? Okay, now remember, these pili are made up of special proteins. Made up of special proteins. Okay, they are extension of plasma membrane, but still made up of special proteins because they need to be hard and they should able to attach to something, correct? They should able to attach to something. Clear? Clear?
Yes.
Have you completed writing?
No, ma'am. Just wait later.
Okay.
Done.
Done? Okay. So now the next structure, we will talk about pili a little more. But before that, we will study one more structure called as fimbriae. Now, fimbriae are like, suppose this is a bacteria. Okay. This is a bacteria. Okay. It is having a small hair-like structure. Okay. Not cilia. Cilia are present only in eukaryotic cell. They're small structures like, what to say? Mm, Uh, I'm not getting correct word to describe them, but small structures, and whole body is covered by these small structures called as fimbri. Okay, fimbri, and this fimbri, again help in attachment. But when we consider fimbri, particularly disease-causing bacteria have them. Disease-causing bacteria, infectious bacteria, have them more. Okay, infectious bacteria. Let's say it properly. Infection-causing bacteria, disease-causing infectious bacteria have them more. Okay, and they help in the attachment of bacteria to host body. Correct. The disease causing bacteria, suppose it need to cause disease in the patient's body, it need to remain attached to the patient. Correct. Okay, it need to remain attached to the person. Okay, patient's body. That's why fimbri help bacteria in that. To host means you can consider patient right now. Host body. Understand right? Clear about it?
Yes.
So this is about different structures we see in the prokaryotic cell. Easy? Easy?
Yes.
Yes. Just many new terms which you have not studied before, correct? Pili, fimbri, flagella, then ribosomes, mesosomes, correct? Don't get confused in this terminology, okay? Start studying, start studying, try to recollect these terms, okay? Try to by heart few things, because you need to give school exams also, correct? In 11th standard, there you need to write question and answer, correct? So you should able to, I'll give you some question answers to write to both of you, to you and Shrishti. So it will be, it will be helpful for you. Now we need to study a very important part called as gram staining. This you are going to do in practical. Okay, see, you will not study that in textbook as such. Okay, but in your practical, you have this practical to do. Okay, and practical based questions come in exam. Okay, practical based questions are in exam. So you should able to do practical questions also. Okay, so let's do gram staining. If you see your textbook, it is just mentioned, okay, gram staining, but procedure is not explained. That is there in your practical book, okay, and you are going to perform that practical. Okay, it is very easy practical, very fun practical to do. You always do, means students always do it with curd bacteria, which is gram negative, okay. But if you do it with gram positive bacteria, results are very, interesting to see, okay. So try to understand what is gram staining. Now see, remember, this gram staining, ये procedure है. What is it? It is a practical procedure, right? It is a practical, literally. You must have not done practical yet, correct? You don't do practical till 10th standard, right?
No,
Okay.
ma'am.
You do? Which practical
Yes,
you
we
did?
have lab. Yes, we have lab in school.
Napa's lab is very good, no? Yeah, I heard so. So which practicals you did?
Like chemical equations. Then one lesson was that animal planted. Teacher shows various types of species.
Different species you have seen,
Yes.
You must have seen
Yes.
amoeba under microscope.
Yes. Also onion peel.
Onion peel cells. That practical you have performed, right? So you used microscope. Have you learned adjusting microscope?
Yes, ma'am.
It is very interesting to learn. Okay.
Yes.
When we were in school, no, one of our friends, he was from Milagris that time, okay, my dad's friend. He was having his own microscope, when we were in school. So we used to, do check practicals with him. Okay, so let's come back to the point. So it is a practical procedure. And this gram in this name, gram, it is a person's name. Okay, it is a person's surname, a scientist's surname. His name was a scientist called as Christian Graham. Okay, he discovered that technique. Okay, that's why it is called as a gram staining. Okay, remember this. Okay, so ग्राम स्टेनिंग में जो ग्राम है इट इज अ नेम ऑफ पर्सन अ वेरी फेमस biologist from Netherlands. Okay, yeah, Danish means the people from Netherlands. Okay, so he was a Danish microbiologist. Okay, so he discovered this technique, and remember, very importantly, this term is very important. It is differential staining technique. Gram staining is what? डिफरेंशियल स्टेनिंग टेक्निक. डिफरेंशियल स्टेनिंग टेक्निक का मतलब होता है, ट्राई टू अंडरस्टैंड व्हाट एग्जैक्टली इज differential means it can categorize bacteria into two different types. Okay, means I will apply same procedure, but I will get some bacteria purple color and some bacteria pink color. Okay, means I can classify bacteria. If some bacteria after my procedure of gram staining, they attain pink color, and some bacteria after my procedure they attain purple color. Understanding? So I can differentiate bacteria which stain purple after the gram staining procedure, and some turn pink. So I can differentiate between the bacteria using this